Okay, first up we've got transcription and translation. So number one, which process is being shown here? I see that there is a ribosome. This should be a big indicator of what process this is. There are also amino acids that are kind of being made into a chain, and there's mRNA. So what's happening here is that mRNA is being read by the ribosome and making a chain of amino acids, which is a protein. So the process that makes a protein is called translation. So that answers number 2. What does the process you mentioned above do? It does not make new DNA. There's nowhere, DNA is nowhere to be found here. It makes mRNA. It actually does not make mRNA. mRNA is only being read in translation. Fixes proteins? No, we didn't talk about fixing proteins in this unit. Last one, it makes a polypeptide. A polypeptide is another word for a protein, and this would be the correct answer. All right. Number 3. Which polypeptide chain, so they're asking about a protein here, protein, is coded by the following mRNA sequence? So they gave me mRNA. I know this is mRNA because it has U's in it and no T's, no thymines. So how I need to use this is separate into codons, groups of 3 bases. So 1, 2, 3, 1, 2, 3, 1, 2, 3. So I know that by the end, I should have 1, 2, 3, 4 amino acids. So A, U, G is my first one. A, so it's in this row. U, this column, this row where they cross is right here. G, AUG is this one, methionine. So methionine is my first amino acid. There, methionine is my first amino acid. That one's done. CAA. C is my first, so it's going to be in this row. A is my second one, so this column where they cross is right here. And A is this third, glutamine, so GLU. That's my second amino acid. Third amino acid, ACU. A is my first base, so it's going to be in this row. C is my second base, so in this column, so where this row and this column cross, right here. And U, U. So threonine, THR. That's my third amino acid. Last but not least, UGG. U is my first base, so it's in this row. G, so where these cross, so right here. UGG, tryptophan, TRY. And those are my 4 amino acids, the polypeptide chain. So this is my protein made by this mRNA sequence. Next up, boom. It says below is a DNA codon. I know that this is also DNA because it has thymine. Which of the following DNA changes would change the amino acid you coded for by the above DNA codon? Hint, you need to use the amino acid chart. So you need to use this chart and figure out what amino acid this codes for. Again, to use this chart, you need to be in RNA. We are in DNA right here. So we need to change this to RNA. And that is A means U, G means C. Whoop. Let me start that again. A means U, G means C, and T means A. This is my RNA sequence from this amino acid. So if I go to my table, U, C, A is serine. So this codes for serine. So I need to find one that would change the serine to something else. So all of these are DNA, right? So it tells me that these are DNA changes. So I need to change each one of these into RNA and then use my chart to figure out which amino acid they code for. So RNA for this one would be U, C, U, U, C, C, U, C, G, U, U, A. So now I need to find each amino acid in the chart. U, C, U. U, C, U. That's still serine. U, C, C. U, C, C. That's still serine. U, C, G. U, C, G. That is still serine. A, T. A, whoop, no, not A, T. U, A, sorry. U, A. That's leucine. So if my DNA changed from AGT to AAT, is the only one that would actually change my amino acid. So D is the only one that changed my original amino acid from serine to leucine. All right, moving on to number 5. Which process is shown below? I see that I have RNA and DNA. So in this picture, I am going from DNA to RNA. And this process is called transcription. And what is created during transcription? This RNA strand right here. So that answers number 6. It makes new mRNA. It makes new mRNA. It does not make DNA. It uses DNA as a template. It doesn't fix proteins, so we didn't talk about that in this unit. And it does not make a polypeptide. It doesn't make a protein. That is translation. So transcription makes mRNA.